Okay, so now we have the last talk of the session, and it's going to be a very exciting one. It's my pleasure to introduce Alison Bentley. She's going to talk to us about data-driven solutions for global wheat improvement. Alison is the director of CIMMYT Global Wheat Program based on Mexico. Her research combines genetics and genomics to develop and deliver new tools and technology to improve plant breeding, crop production, and adaptation to climate change. At CIMMYT, Alison leads a team of scientists using scientific approaches to develop improved wheat germplasm. And this germplasm captures packages of traits providing productivity, resilience, and broad adaptation, which supports global wheat improvement. Prior to joining CIMMYT in November 2020, Alison worked in the UK focus on translation of fundamental scientific breakthroughs into tangible impacts for the agri-food sector. She has a doctorate in agricultural science and a PhD in agriculture from the University of Sydney in Australia. Alison, please go ahead.
Thanks, many thanks for the introduction, and thanks to the organizers for the opportunity to present work from the CIMMYT Global Wheat Program. So I'll start. Hopefully you can see my screen. Let me know if not. So just for a little context, although we've already heard a lot about the importance of crop species, wheat is essential to global food security and really in the news at the moment with the situation unfolding in Ukraine. So in terms of the production and CIMMYT's mandates, over half of the world's wheat production is in developing countries, and this is really the core of our focus at CIMMYT. And we know meeting future needs, that there's a huge gap between the projected supply and the projected demand, but we also know there's some inequality in the distribution and the magnitude of the increase that's likely to be needed. And this is specifically focused on sub-Saharan Africa and South Asia, where the need for increases is a much higher rate than the rest of the world. And this is part of the challenge we try to address through wheat improvement and distribution, supporting producers in Latin America, Africa, and Asia to boost productivity and support livelihoods. So just to summarize what we do within the CIMMYT Global Wheat Program, a lot of work using data-driven methods. I guess everything is data-driven these days to a certain extent. So we go all the way from genetic resources through elite germplasm, tested across multiple international environments, using and incorporating the latest breeding tools and technologies that are available, which includes phenotyping methods, all the way up to satellite observations. And then at the more genomic level, using marker-assisted selection, pangenomic data, and haplotype-based selection methods, along with genomic selection, and then also focusing on what the end products are and what the quality traits are that are needed to achieve quality traits from wheat, which is a global commodity used to create a very wide range of products in the food and feed sector. So just to give a quick background on the CIMMYT wheat breeding program. So this has been established for over 50 years here in Mexico. So we run a shuttle breeding scheme, which takes advantage of the different geographies within Mexico and allows us to run multiple breeding cycles each year by moving material between El Batán and Toluca, where we can screen intensively predominantly for disease pressure, and then to Ciudad Obregón in Sonora, where we can select under high yield potential in irrigated environments, but also screen for water use efficiency, heat tolerance, as well as some additional diseases of relevance. We added a shuttle to Enduro in Kenya when stem rust broke out and became a threat to global wheat. And this allows us to add an additional phenotyping step for stem rust, which is a pathogen of growing global importance. So here in Mexico, we breed wheat for the world, so really targeting South Asia, Africa, West Asia, North Africa, as well as Latin America. And this has proved to be hugely effective, running this centralized breeding program from Mexico, taking advantage of the ability to run multiple breeding cycles in a year in these diverse production environments. And you can see here in this graph on the right-hand side, the material produced by the program has very high documented rates of uptake in our target geographies, either as direct releases, so the line that's bred here in Mexico is directly released into these environments, or used as a parent in the breeding process. So this is important because it means if we can get a genetic innovation into a CIMMYT variety, we know we can deliver it through our breeding strategy to customers, our stakeholders, farmers, throughout many diverse regions of the world. So as I mentioned, we're breeding here in Mexico for the world, and our focus areas are shown here, so South, Central and West Asia, Southern and Eastern Africa, and Latin America. And each of these dots here represents testing environments for the material produced here in Mexico and distributed to partners in these different geographies. And as I said, this centralized breeding strategy has for a long time proved very effective, and it's obviously a good way to centralize your resources as well. And we can track the breeding progress over time. So if we look in our selection environments that we use in Ciudad Obregón, we can see a continual grain yield progress over time in our breeding material. So at our breeding station, across simulated environments for high productivity, which is the green line on top. So that's a fully irrigated wheat production environment. And then the two lines on the bottom that are brownish and purple show the performance over time in drought and heat stress, so stress adaptive trials. So we see this consistent increase over time. And then if we look at our target populations outside Mexico, we can also see this continual increase in grain yield potential over time, in this case in India. So we know that this centralized breeding strategy is very effective at having an efficiency of scale, an efficiency at scale that allows us to deliver these gains to partners around the world. And we've recently done a more granular analysis of these rates of genetic progress, which show significant progress over time in multiple environments in South Asia and West Asia, which are shown here. So as I mentioned, this material, which is bred here in Mexico, goes to partners all around the world and generates a huge resource and source of data, because data is also collected on the material and returned to us to be used to inform future breeding decision making. So this is a huge scale, over 90 different partners participating in this network on an annual basis and returning data that's then used to inform breeding decisions. So we know that over time, the programs delivered accelerated rates of genetic progress, which is really what we quantify in plant breeding programs. And now we're really looking at how we accelerate that even further. And we're doing this in a number of ways. Some of them are infrastructure or kind of technology based, and others are genomic or genetic based interventions. So faster breeding, we're using new greenhouse infrastructure, which we've built in Toluca, where we can use semi-controlled environments to reduce the breeding cycle time from 6 to 3 years, still taking advantage of the ability to shuttle material between central and northern Mexico, but speeding up the earliest generations of the breeding process. We're also using genomic selection, which Sarah mentioned in her talk, for advancing material through the program and recycling parents back into the start of the process. And then we're using speed breeding, which is shown here on the right-hand side of the graph, of the slide, sorry, to introgress novel traits, so we have genetic markers available or a new variation detected for rust or disease genes or QTLs of interest. So this just gives you an idea of the scale of our accelerated breeding infrastructure, which is now operational in Toluca, and invite everyone to come and visit us and to see this in action. So here we have over 2 hectares of covered space, which, as I mentioned, is a semi-controlled environment. And really the idea here is that we can move more quickly through the early generations of the breeding process. So we've recently been making lots of crosses, so over 1,000 crosses, which is shown here on the right-hand side, aiming to really speed up the practical component of the breeding process. And why do we do this? We do this because simulations show that using these more rapid methods, obviously based on quantitative genetic theory, that the faster you go, the more genetic gain you can make over time. But also aware of the effects of that on genetic variants and strategies to also fill or maintain genetic variants over time. So just to move on, really, I think we're really focused on genetic improvement, genetic progress over time, and targeting that to a really broad geographical range that the CIMMYT Wheat program delivers to. And using genomics is a large part of the advancement of the program. And so the wheat genome is highly complex and is something that is quite challenging to work with. So in Kevin's talk, he talks about a lot of diploid species. In the case of wheat, we have a huge magnitude change from something like maize or corn, 2.5 gigabases to wheat, which is it's a much larger genome and is also a hexaploid. So for those who aren't familiar with wheat, it's a hexaploid species which combines in its genomes the genomes of 3 ancestral grass species, which came together over 2 successive hybridization events in evolutionary time. So the first brought together the A and the B genomes around 100,000 years ago, leading to wild Emma wheat Triticum dicocoides. And dicocoides has been subsequently domesticated into Triticum dicoccum, Triticum durum, the wheat that's used to produce pasta and semolina and other products. And then the second hybridization event brought in the D genome from a wild goat grass called Agelops tauschii. And this happened much more recently in evolutionary time, so around 10,000 years ago, and brought in a lot of new diversity. And much of the gene bank mining work is really about revealing the diversity and novel functional genetic variation from these goat grass wild relatives. So it's very significant in terms of the size of the wheat genome, but also the complexity in terms of its hexaploid constituents, which creates mostly issues for cloning of genes, creating gene-based markers for use in the breeding process. But the progress of wheat genomics has been very, very fast. So when I started working in developing new markers in 2007, we had a handful of biparental mapping populations and SSR markers, which were very laborious to run. And just over 10 years, really, now we see wheat can be used, is both a global food security crop, but is also a really interesting species for doing functional genomics for cloning genes, for understanding structural variation, and for doing a lot of fundamental science in a crop species. So now we have within the community a lot of germplasm resources, as well as a lot of genomic sequence information. And this is really changing the game, not only for the scientific community, but also for the breeding and improvement of wheat, because all of this technology can now be deployed to help breeders make better decisions faster. So this just shows you within the CIMMYT Global Wheat Program, the integration of genomic tools, which has been a very active component of the program. So very similar to what I just presented from my personal experience as well, is around 2007, we were really at biparental mapping, the start of the GWAS era, marker-assisted selection with kind of complex acrylamide gel-based markers, through to today, where we're using these rapid generation advanced facilities, have fully implemented genomic selection on tens of thousands of candidate lines in the breeding program, and have centralized genomic genotyping services which can deliver data, additional layers and additional complexity of data to breeding programs. And part of that complexity is really understanding what we do with all of this genomic and genetic information we now have access to. And this is really an ongoing process for us as part of the breeding program optimization. So in a breeding program, you have many different stages, from your development of your parents, improvement of the population, selection throughout generations, advanced testing, and then the recycling of parents. And you can use genomic data at any of these stages, which makes it both good and bad, because you need to work out where and when to use the genetic or genomic information that you have access to. And this just visualizes this in the same way. Really, we have this huge resource of information, and are now at this stage really trying to map it into the stages of the breeding program where it can have the greatest possible impact for improving decision making and the material we produce. So one aspect of this is trait augmentation using a speed breeding facility, which you can see here in one of our bodegas at Toluca Station. And this is using heliospectra, LED lights, continuous lighting to try and incorporate a number of target traits or genes that we need to have available for our wide range of geographies that we serve. So many of the farmers that we deliver material to don't have access to agrochemicals. So the variety or the wheat itself is the first line of defense against a major pathogen or pest outbreak. So within the program, there's really a strong focus on disease resistance. A lot of work has been done both in cloning our genes and also cloning adult plant resistance genes, and then looking at the strategies to deploy those through forward breeding. So in our speed breeding pipeline, we're now using marker-assisted backcrossing, relatively straightforward to try and create improved material which carries all of the necessary genetics that we need at the end of our breeding process. So this includes striped frost stem rust, Fusarium headlight septoria, and we're also testing the use of speed breeding and marker-assisted backcrossing to incorporate novel haplotypes identified from gene bank material. So this is a relatively straightforward operation where we have markers tagging each of these genes of interest or PTL of interest and want to introgress them into elite material, but quickly generates large numbers of plants and data points, and does it in a very short period of time because we're using speed breeding, which allows us to move through 4 to 5 generations a year, which means we have a lot of associated logistics challenges as well. And this has led to the need for genotyping workflows, which are now in place. So we're able to sample the material, extract the DNA. We do a lot of the genotyping services outsourced, and then data return into a data storage system that allows us to process and use this data in real time to make breeding decisions. So this is really important, being able to generate data not only for research purposes where the timelines are less critical, but for fitting within these multiple generations of the breeding cycle each year. And we've been working with the Excellence in Breeding platform, which is across the CGIAR system, to develop a new mid-density genotyping array for the routine implementation of genomic selection, and this just shows you the genomic distribution of the markers on that array. So it's the eternal search for optimal genotyping platforms and solutions that are not only descriptive of the material and allow for the making of predictions, but that are also fit within the logistics of a live breeding program with hundreds of thousands of individuals being progressed through multiple generations each year. So at Summit, we within the Global Wheat Program, we have big data, but by that we mean we have a lot of data genotyped with different platforms and a huge amount of potential information to mine. but sometimes the challenge of a lack of contiguity between these data sets and the eventual uses in breeding. So in forward breeding, we still rely mostly on Cas markers, so tagging specific genes, alleles of interest. And then as Kevin described on the gene bank, you've got high density genotyping by sequencing on thousands of elite lines. And really the challenge is how we unify all this data and use the data sources to make informed decisions for taking material forward into the breeding process. So if we look at the wheat genome, the post-genome sequence era, which we're finally in for wheat, this huge and highly complex genomic beast, we can see that we're really now at the stage of being able to understand more about structural variation and haplotypes, leveraging the investments that were made by the community in creating core and pangenome references. And CIMMYT's contributed to these significantly, so through the sequencing and assembly of several wheat genomes, contributing to the pangenome effort across the wheat community, and also the lower coverage sequencing of material from the breeding program. And then a huge amounts, as I said previously, of more genotyping by sequencing, high density genotyping data on lots of on hundreds of thousands of lines from the breeding program. And also alongside these resources to understand the genetics, we also have huge catalogs of QTLs which have been discovered across multiple projects, across multiple time points. And really now the challenge is we've just created this QTL catalog to catalog all of the QTLs which have been identified over time in multiple populations against the reference genome. And really the challenge is how we use all this information to inform breeding. And really the focus at this time now, we have this vast, well, probably not vast in the Arabidopsis sense, but we have really for the first time pangenomes. We're going from sequence to haplotype, and this is a really big focus now to move forward on how we use haplotypes, which are likely to be of greater interest in terms of some of the complex trait phenotypes we're looking at in the program, like drought and heat resistance, and much more complex traits. And so we have a strategic partnership with the John Innes Center, so a lot of this work is part of that collaboration. And this just gives you a snapshot of Borlog 100, a CIMMYT variety aligned to Jagger, one of the pangenome sequences. And here we're trying to identify divergent haplotypes or haplotypes associated with potential functional variation for use in forward breeding. And you can see here also we're able to use the tools that are now available for visualizing and characterizing haplotype variation. So really keen to take this forward. And if anyone has an interest in working in this area with us, there's a huge data resource, and then our ability to validate this actually in the breeding context allows us to scale up from the comparisons of pangenomes and the identification of putative functional variation into breeding trials distributed across the world. And that's really the power of combining genomics into a real, very large scale global breeding program. So I'm aware the time is short. Apologies if I went over time, but very happy to hear from anyone who wants to hear more about the Global Wheat Program. And thank you very much for your attention.
On wheat improvement. Is there any questions for Alison?
Hi, can you hear me? I have a very basic question. The lines, the wheat lines that CIMMYT releases, are they released as varieties that the farmer can directly use, or are they released as breeding lines for local national programs. Because at least in the case of corn in Mexico, most of the varieties that are used come from commercial sources, not from CIMMYT or national programs. So what's the case for wheat?
Yeah, thanks for the question. And obviously wheat and maize are quite different because of the wheat as an inbreeding species. So we provide to our partners, which are national programs around the world, sets of advanced material, and those are often directly released. So it really depends on the context. So the material is at a sufficient level of improvement to be directly released. So in the context of somewhere like Yemen, where there isn't a functioning national breeding program, the material can be directly released and often is. Through to countries like India, where the material goes into assessment alongside as part of the national breeding programs that exist there, and is then used as a parent, or also could be directly released. So it very much depends on the context, but the aim of the program is to deliver material that has the full package of traits, so that if it is directly released, then it's directly released through the national program, or through a linkage between the national program and a private sector partner, depending on the modality in that country.
Thanks, Alison. Hi. You mentioned UG99 at the start of your talk, and I remember reading a few years ago that it was a major threat for the wheat production in the world, that it was like going from Africa to the Middle East and to Europe. I lost track of what happened to that infection in the world. So what's going on with that UG90 data? And there's any breeding being conducted for resistance to this UG99?
Yeah, definitely. It's a great question. And I think when diseases fade from the front page of the newspaper, it often doesn't mean they've gone away. It just means we've found a solution. And that's very much the case. So we really invested a lot in ensuring the material that we produce has resistance. So all of the material that comes out of the program has a threshold level of resistance to stem rust. And so we do that in partnership with Calro in Kenya, where we've established a platform for screening stem rust. So all of the material from our program here in Mexico will go to Kenya and have an additional round of field phenotyping for UG99 for stem rust and the races that have evolved since the outbreak of UG99, because rust pathogens are continually evolving. So that's a really important component of the program, as has been the identification of sources of resistance and how we pyramid those and make sure full complements of them are deployed as part of our breeding strategy. So any material we share anywhere in the world, even if it's not in a stem rust area, will have a baseline level of resistance to stem rust. For exactly the reason you mentioned, that the pathogen can spread to new environments, new geographies, and so we have that as a base criteria for the material we produce. And then the other side of that is obviously the surveillance, and so we do also have Dave Hodson and various others at CIMMYT who are running the active surveillance for stem rust, as well as for the other rust pathogens specifically throughout Africa, South Asia, and that monitoring and surveillance is also really important for knowing when and how the pathogen's changing. And I think later you'll have a talk with Sophie and Kamoon, also using genomic sequence information to track pathogen changes over time. So that's kind of the other part of the story, understanding how the pathogen's changing, and then having a way to screen tens of thousands of candidate lines to make sure there's sufficient levels of resistance in the material. Thank you.
Thank you. So any other questions? If not, let's thank Alison again for a great talk.